So we will be starting with the chapter Molecular Basis of Inheritance and the topic Search for Genetic Material. Before it was proved that the DNA is the genetic material, there was many scientists, they worked and they performed many experiments and proved that DNA is the genetic material. And before that, this performed experiment to find out whether the genetic material is DNA or RNA. So during that course of experiments, one experiment performed by Frederick Griffith in 1928, he performed experiment with the mice and bacterium Streptococcus pneumoniae. And during the course of experiment, he observed that the bacterium produces two types of colonies. One is S strain, he called them S strain, and other is R strain. On the basis of the smooth coating produced by the bacterium, so the S strain bacterium, they produce smooth colonies, because of production of mucous membrane, mucous coating, and these S strain were virulent, means, or we can say they were pathogenic. And R strain did not produce any smooth colony. In fact, they produced rough colonies, and these were non-virulent or non-pathogenic, did not cause any disease. So S strain caused disease in the bacteria. So what was his experiment? What he did, let's talk about the steps of his experiment. Basically, in the first step, he injected R strain into mice and observed that mice survived. Second step, he injected S strain into mice and observed that mice died because of the disease. Because S strain were virulent, they caused disease and mice died. R strain non-virulent, injected into the mice survived. Third step, he killed S strain by heating them, and then he injected heat killed S strain into mice and observed that mice survived. Point to be noted, when he injected S strain, normal S strain into mice died. When he killed S strain by heating them and then injected into mice survived. Now in the fourth step, what he did, he mixed heat killed S strain with the R strain. So the mixture he injected into the mice. So what he took heat killed S strain with R strain and injected into mice and observed that mice died. See, when he injected only heat-killed S strain into mice, mice survived. That means heat-killed S strain did not cause any disease in the mice. So mice survived. But when he injected normal S strain, mice died. Heat-killed S strain, mice survived. But now when he injected heat-killed S strain mixed with the R strain and injected, and mice died. That means there was something happened, or we can say something from heat-killed S strain transformed R strain into virulent. So the mice died. That causes disease in the mice, and mice died. So this was the transforming. That's why this is called transforming experiment. And he concluded on the basis of his observation and experiment, that there was something which transformed R strain into virulent, and the transforming substance was genetic material. But he did not prove that the genetic material is DNA or RNA or protein. But yes, he proved that there was something, or that something was the genetic material from the heat-killed S strain transformed R strain into virulent. Now, this was his experiment only. Later, every three scientists, MacLeod and McCarthy, they together performed, experimentally. They together experimentally proved that the transforming substance was the DNA. DNA was the substance which transformed R strain into virulent. So what was their experimental proof? To prove that, they extracted every MacLeod and McCarthy, three scientists, every MacLeod and McCarthy experimentally proved that DNA was genetic material. During Griffith experiment. So, how they proved experimentally? For that, they extracted DNA, RNA and protein from heat killed S strain and took these mixtures into three different test tubes. So 1, 2 and 3, he took DNA, RNA and protein, same in all these three test tubes. All these three test tubes, he took these mixture extracted from heat killed S strain. Now in the first test tube, they added proteases and injected this mixture to the mice, and mice died. That proved that it is not the protein, because adding proteases digested the protein from the mixture. So there, in this mixture there is no protein, and when this mixture was injected along with the R strain. So this mixture along with the R strain. So in all the three test tubes, they took DNA, RNA, protein extracted from heat killed S strain plus R strain. He added R strain, all these three. So these are the things he took in these three different test tubes. In first test tube, they added proteases. So the proteases will digest protein from the mixture, and then this mixture injected into the mice and mice died. That proved that it is not the protein which transformed R strain into virulent, because when they digested protein, even then the mixture is, there is something which is transforming R strain into virulent. Now in second test tube, they added RNAs along with the DNA, RNA, protein plus R strain. In this test tube as well, he added RNAs which digested RNA. Now this mixture injected into the mice. They observed, again mice died. That proved that again it is not the RNA, because from this mixture they digested RNA. Now in this mixture there is only DNA, protein and R strain. Again mice died after injecting this mixture into the mice died. That proved that it is not the RNA, because if it is the RNA, after digesting RNA, it will not cause disease. So it proved that it is not the RNA. In third test tube, they added DNase, enzyme, to digest DNA now. Now in this test tube, there is only RNA, protein and R strain, and when this mixture was injected into the mice died. Right? So, mice survived. And in this, there is no DNA, because after adding DNA digested. So in this mixture only RNA, protein and R strain. When this mixture was injected into the mice survived. That means it was the DNA. It was the DNA, because in these two mixtures there was DNA. So it was the DNA only, and in this mixture there was no DNA. No DNA, and when this mixture injected into the mice survived. That means it was only DNA which transformed R strain into virulent and caused disease. So this was the experimental proof given by Avery, MacLeod and McCarthy that the DNA is the genetic material, and this was the transforming principle for Griffith experiment. So that was all about this topic.